All right, we are live. Cool. Well, thanks for joining us, everybody. This is another installment of MarshChat, where I talk to awesome marsh ecologists about their research and about salt marshes in general. Today, I am really excited to talk with Rebecca Atkins. Hey, say hi, Rebecca.
Hey.
Rebecca is a graduate student at the University of Georgia, and she is doing really interesting research on size diversity of different salt marsh consumers, specifically one of the most important in the Southeast, the snail Literaria irorata. So Rebecca, it's great to have you. Thank you so much for joining me.
Thanks for having me on, Mark.
Of course. So we'll get started today just kind of talking in general. So Rebecca, you are a different type of marsh chatter. You're what, in your second year of your PhD? First?
I'm in my first year.
First year of your PhD. That's awesome. And so tell us a little bit about why you study salt marshes. Why do you think studying salt marshes is important?
Well, salt marshes are one of the most productive ecosystems in the world. And I also started working on salt marshes when I was a sophomore undergrad at the University of Florida. So I kind of got hooked. It was my first research experience, and I've just enjoyed spending time out in the marsh. And I think that they're really valuable, both from like an ecosystem services perspective and just for their natural beauty. So I hope to continue working in them.
Yeah, totally. So one of the things that that I love about marshes, especially like in the Southeast, is how many, but basically just the kind of massive abundance of species, right? There's lots and lots of crabs, lots and lots of snails, but you know, overall diversity isn't very high in like number of species, right? But something that that you see when you walk into a southeastern salt marsh is a huge diversity of sizes of things, and that's what some of the stuff that you're really interested in, right? Tell us a little bit more about that.
Yeah, so something that initially caught my attention when I was looking in these, the marshes on Sapelo in particular, were the differences in sizes within literaria. So in other words, you have small snails, you have large snails, intermediate sized snails. And you can kind of see this like there's a zonation within site in terms of body size. And then I'm also interested in kind of the differences among sites as well. So I think that literaria, I think that different factors could potentially be driving size structure within these literaria populations. And I'm hoping to look at exactly what that might be, if it's more abiotic or biotic species interactions or environmentally driven. And then what that means for how they feed on the Spartina and that relationship between literaria and productivity.
Awesome. So I'm going to try here this screen share, which I've been working on, so we can talk a little bit more specifically about some of these things. So to give a little intro to basically what these snails look like. Oh, you know what? I don't know. Wait, did you send me that picture of the snails of different sizes?
I did send you, yeah,
Okay.
mostly the snails that they're laid out on like a little tinfoil sheet.
Okay. Hang on, let me grab that real quick. I actually don't.
Oh, okay. I can send that to you right now, actually.
Yeah, send it to me right now, where I pop up basically a picture of a big snail. And hopefully this works. You'll have to tell me, because this is my first time doing the screen share. Did it pop up on my side?
Yep, I see it.
All right, yeah. So this is the snail in question, the Literaria irorata that Rebecca's been focusing on, and she's going to give me a picture of the different sizes of them. But these guys do some pretty unique things in salt marshes. Specifically, they make these cuts on the grass right here. This is a cut in the Spartina grass. And as you can see, this cut is infected with this brown black stuff that's fungus. The snails actually farm this fungus. They poop in the grazing marks that they make, which have fungal spores in them. Their feces does, and in turn they actually end up eating that fungus, right? So, but something that that we've found throughout our research in the Southeast, Rebecca as well, is that these what these snails do in the marsh actually has a lot to do with their size, right? So here's another picture of the snail grazing on some marsh rats, or maybe this guy might just be crawling up. But so let's see. I'm gonna switch back over to Rebecca's screen here.
Yeah,
oops, that's on me.
That picture.
Yeah, okay, cool. And I'm gonna grab that other picture. So Rebecca, tell us a little bit about what you know about what different size snails do. What do they do when they're small, medium, large, that sort of thing?
Okay, yeah. So actually, the funny thing is that the work that I did on Sapelo when I was an undergraduate suggested that it actually may not really matter in terms of body size of these, or like density of these snail populations per se, and that it actually is just a function of the energetic demand or metabolic biomass of these snail populations. And initially I was surprised because I anticipated that there would be a difference between small individuals and large individuals. Maybe they would have different feeding preferences, or perhaps smaller individuals would have maybe a less developed radula and be unable to graze or leave those radulation scars on plant tissue. And in turn, like they would need to reach a certain density before they were able to really have a consumptive effect on cordgrass productivity.
Right. So yeah. So hang on. I'm going to make my screen big again. Okay. It should be. This is a picture of the grass again, right?
Yes, that is
okay. Cool. Yeah. So like Rebecca is saying, right. This is a radulation that bigger snails make, right? Like, what is there a minimum size of snail that can make this? Or you might not know, but just like a guess. Do you think?
Yeah. So I'm not entirely sure, and that's something that I'm contemplating looking at this summer. I tried doing feeding trials using different sizes of litter area, and it's really hard to get them to actually graze when you put them in a mason jar with a blade of Spartina.
Yeah.
So I'm going to try working with the actual Spartina plants in pots and mesocosms this summer,
cool.
and look at body sizes. And also maybe potentially like take a look at the like images of the radula to see if there's any like morphological differences or exactly like how they develop.
Oh, very cool, right? So you thought there would be that like the size of the radula determines when it can start making these grazing marks in the grass?
Potentially, like maybe if smaller individuals are less able to access tougher plant tissues, they could be using the scars left by larger individuals, or maybe a certain number of smaller individuals to get together and graze as like a group. I don't really know too much about whether or not that actually happens or the potential for that to happen, but it's definitely worth looking into when thinking about size variation.
Cool. So here is the picture of all of the different sizes of snails here, right? And so we've got large snails up top. Maybe you walk us through this, Rebecca.
Yeah. So this looks like, I'm trying to remember the exact site this was. It could have been the Yellow Trail site in St. Augustine, Florida. But essentially, this is just a picture of one site's worth of snails. So I collected them in quadrats this past summer just to look at general size, structure, biomass, and densities of snails within 1 meter square quadrats at 12 different sites along the East Coast. And so this looks like it could be from a site in St. Augustine, Florida. And so you can just kind of see you have just a myriad of sizes, large and small individuals. It's probably hard to see the really tiny ones, but some sites have, the size structure is actually different among sites. So some sites I work at have predominantly large snails, some have predominantly small snails. So Sapelo Island is a good example of a site where I find a large amount of smaller scales of snails relative to my other sites. There's a site really far north where I just have predominantly large snails. So size structure varies and within and among sites, essentially.
Yeah. Okay. Cool. Then, yeah, I mean, it's a striking thing when you like really start to put your head in the grass, so to speak. You start seeing actually. Oh man, that would have been a perfect time for this pun. Hang on. Give me 1 second here. Yeah. When you put your head into the grass, like for instance, Rebecca is doing in this picture. Did that come up well?
Excellent.
Oh, perfect.
Good
right.
time.
So when you really put your head into the grass, like this is one of the things that that's super striking, right? Is that like there's a bunch of different sizes of these snails, and as you're saying, the bigger ones are the ones that we know can make those slices in the grass. But then there's like medium-sized snails. There's tiny snails, right? I mean, what do we say that? What's the tiniest snail that you've collected?
The tiny. I mean, usually I'd want to say it's like I'm trying to think of like something you can use for size reference. It's definitely smaller than like a typical pea. It's like maybe a couple millimeters.
Yeah, like the height basically of the snail, like a couple.
Yeah, like 2-3 millimeters, and those are the ones that I'm able to see and collect. But a lot of times the recruits will come in and they can hide themselves in the furled blades of those Spartina plants, and it's hard to get to them.
Yeah, that's and that's one of the most amazing things, right? Is these snails, when they reproduce, right? They send their larvae out and then they settle, basically as like the tiniest little pebble of a snail. And then, they got to start eating something, right? And they're not, like Rebecca talked about, they're not strong enough to start grazing on the grass. So what is it that they eat when they're that small?
It's possible that they would graze on like the benthos, diatoms, epiphytic algae that will grow on the stalk of the Spartina. And that's another thing that we're thinking about measuring this upcoming summer, because you can put out straws even and actually measure the algal growth on these straws and just see exactly what the what food may be available to littoraria at different sites.
Very cool. Yeah.
So,
and then, so Rebecca, tell us a little bit more about the experiment that you published from your work on Sapelo. Was that 2014 that came out?
It came out in the beginning of 2015, I think, actually, like officially it was released in 2015.
Yeah, so this was a really cool experiment, right? Where you manipulated the sizes and density, or the densities of different size snails, right?
Yeah, so we had different size classes, large and small, and we manipulated densities. I think we had 5 different densities ranging from 35 to 300 individuals, if I remember correctly. And we were just interested in looking at whether or not you could parse out the effective size and density on grazing intensity. And we measured, grazing in the form of radulation scars, and we also measured it in terms of above ground biomass of Spartina remaining at the end of the growing season. And what we found is that you could pretty parsimoniously explain the relationship between these littoraria populations that were stocked in these cages and the final above ground remaining biomass using a single linear function. And on the x-axis of that was population metabolic biomass. So it showed us that size and density per se may not be like driving factors in and of themselves, and it's in fact the effect of both of them on the population metabolic demand.
Cool, very, very cool.
Effects. But I would like to keep looking into that, especially looking at size variation among sites.
Yeah, so this picture here actually is this from Sapelo or no? This is not.
No, that's actually that one's also from St. Augustine.
This is St. Augustine, yeah. But these are basically what the cages look like. Actually, one of the really cool things about Rebecca's work is that, a lot of us either include or exclude snails or crabs or whatever it happens to be. But when you're excluding little tiny snails or including little tiny snails, you have to do something a little bit unique, right? What was the size of the mesh that you used to keep these little guys in?
So it's a 1/4 inch mesh, I believe. I didn't want to do anything that could potentially lead to really large caging effects. That really tiny mesh might change the way water's moving and sediment could be accumulating in the cages. So I do go out and have to check to see whether or not snails are actually being excluded. And for the most part, I think it's doing a pretty good job.
Yeah.
Galvanized hardware.
Yeah, it's definitely those little tiny, obviously, they don't affect flow, but they gotta keep the little snails in, right?
Right. And so, what I have out right now is mainly snail exclusion cages and then plots that are just open to grazing. And so right now I'm just trying to get an understanding of how the population of littoraria as a whole within each site is affecting Spartina. Is it having a positive effect, a neutral effect, a deleterious effect? And then once I am able to kind of get a better understanding of that, maybe dig deeper into what exactly it is that's causing size structure variation and the implications of large versus small individuals at each site.
Yeah, that's really interesting. Do you have some thoughts on what that could be? What causes basically the big difference in the sizes of snails out in the marsh?
So what initially motivated me to look at a latitudinal gradient was temperature. Looking at how potentially colder temperatures would select for larger body sizes. So in other words, snails at these northern latitude sites would have an overall larger average body size than snails at southern latitudes. And so in general, based on the sites that I'm working at, that appears to be the trend that I'm seeing. However, I know of sites within these latitudes that also have a large range of size variation. And so I think that there could also be a really important role of predatory crabs and driving size structure as well. And then maybe it could also have to do with Spartina palatability, something to do with nutrient availability. So I'm trying to look at pore water chemistry and how that relates to Spartina growth and palatability. And then also we'll take stock of predators and their size structure as well this summer, just make sure that I'm kind of covering all my bases and really getting a more big picture understanding potentially of what could be influencing snails.
Yeah, very, very cool. So, I mean, if we talk about what sort of predators are around in southeastern salt marshes, we think of mud crabs and 2 different types of mud crabs, right? Blue crabs as well. And then, from work that Nicole Sumzat did in Sapelo, of course, the purple marsh crab eats little small snails too, right? So based on what we know about predators in southeastern marshes, what are your instincts on basically who is eating what when it comes to predators and snails? You have some ideas?
So mostly in terms of like these marshes, what I think is eating the snails, I think a lot of it is due to crabs. So mostly the mud crabs. I see a lot of black clawed mud crabs, and then also a lot of blue crabs. I've actually had blue crabs fall into my cages. So I am seeing them, and I do think that they're having an effect. I've seen marsh rails also, the clapper rails, and so
the clapper rails, the bird. What are they also called? Marsh hens too, right?
Marsh hens, they make crazy sounds. But yeah, I've seen those guys around. But I have noticed, like around some of my cages, especially the ones that where crabs are nearby, I see these little shell middens or little crab middens that have a collection of just dead littoraria shells nearby.
Yeah.
So I definitely think that they're doing a lot of damage and could potentially be influencing shell morphology and grazing behavior of snails at each site.
Very cool. Very cool. So, all right, Rebecca, well, I want to ask some other questions too. I'm sure that a lot will come back to snail sizes, of course. But so what is your favorite life history story from the marsh?
Life history, like my life in the marsh?
Like, no, like an organism's life history story.
Organism's life history story. That's an interesting thing to think about. I mean, I could pick... I don't know, there's so many things to think about. I mean, I feel like I should say snails, but I don't know.
What's the first one that pops into your head?
I don't know, blue crabs. I think blue crabs have a cool story, mainly because I went out this past sampling trip in October and I was expecting to see blue crabs, and not October, actually, it was a few weeks ago. So it was in March, and I told the undergraduate who was helping me, like, I can't wait for you to see these blue crabs. They're amazing. And then we get out there and we don't see a single crab the entire week. And then I learned that they actually would move back out into the ocean when it got colder, and they kind of buried themselves in the mud. And then crabbing season wouldn't start until early to mid-March. So I did not know that they weren't present like year round, that they had more of a seasonality.
Yeah, well, and that totally affects, what times of year that they're in the marsh eating snails, right? Or eating mussels or whatever that happens to be.
Definitely around in the summer. So.
Yeah, very much so. Cool. So what is your favorite marsh? I think you have a cool perspective on this, because part of the stuff that you've been telling us about is, you've been basically surveying marshes, from Florida up to Virginia?
Yeah.
Yeah. So of all those places that you've been to looking for snails of different sizes, what's your favorite one?
Honestly, I love Sapelo Island. I know that's like super cliche, but I think it's beautiful. There's so many different marshes out there, and the Georgia coast, Georgia and South Carolina together make up, I think, the largest stretch of like contiguous salt marsh in the U.S., if not the world. So they have some gorgeous salt marshes, and I like working in South Carolina as well. Some of the northern marshes, they are vastly different from what I got used to on Sapelo. So
yeah. Did you get to spend much time in northern marshes?
So I guess, okay, my definition of northern was like Virginia. I did go to Delaware and Maryland, and I saw those marshes. And I found littoraria, but they weren't distributed in the same way that I was seeing in Virginia South. And so I haven't really included those in this study. But do you see very many of them up in Massachusetts?
No, there's not. There's the littoraria, the Littorina and Littoria, which are around, but they don't do a whole lot. There was a study, I can't remember when it was, but they basically tried to do the same sort of increased snail density experiment and look at the effects on primary productivity. And it takes a lot of snails to even start, grazing on the Spartina alterniflora. They mainly eat the benthic algae, diatoms and stuff like that, which is interestingly, right, the stuff that the smaller snails are most likely eating down in the southeast. But yeah, the impact of those grazers is pretty minimal up here in the northern marshes.
Yeah, interesting. I know Melampus are up there, but I guess they also probably prefer the benthic diet.
Yeah, the Melampus, the coffee bean snails would also just graze sort of on the diatoms on the stems of the plants. Yeah. So yeah, of those, give us some kind of like big picture, like what's the difference between a marsh in Virginia and one in Florida or in South Carolina or in Georgia? Like kind of your just general impressions.
General impressions. Well, aside from the fact that I think there's just more like area of marsh in South Carolina and Georgia, I think maybe I'd consider them to be more, I wouldn't say pristine, but it feels like, you're out in the marsh when you're working there. Whereas some of my sites in North Carolina and Virginia, you can see development, housing. It doesn't really feel, except for the site on the Eastern Shore when I'm on the mainland of Virginia and North Carolina, you can definitely tell that you're surrounded by people. So I think that's going to have a big impact potentially on like species interactions and maybe also diet structure and nutrients going into the marsh, things like that. And so I'm trying to figure out exactly how to incorporate those kind of like metrics into this study, or if it, figure out how it may be playing a role. The marshes in Florida are unique in that you have that mangrove-spartina interface, because that's kind of, I guess, the southern range of spartina and mangroves are starting to expand northward. And so that also kind of adds a new dynamic by bringing in a different species community associated with mangroves. So my sites right now aren't really looking at that interface, but it's definitely something that I think about when I'm there.
Yeah, that's really cool. I mean, that's something that the research is kind of just getting started on that. But yeah, basically, the big mangrove forests, which are these basically trees that have big old prop roots. They are basically moving into the areas where salt marshes are because it's getting warmer. Normally, it's the mangroves are excluded via cold, but since the temperatures are getting warmer, they're starting to move into areas where marshes are. So that I would imagine that would be a very interesting dynamic.
Oh yeah, right now you have a lot of like black mangroves, which have those the metaphors.
Yeah, the metaphors. Yeah.
And so that's like changing the structure and species. There's been some really cool research. Benthics, they had a couple of really good talks on what's happening with that. So yeah, and then you also have just the mainland island gradient. So Sapelo is an island. The mainland on St. Simon's looks a little bit different than it does on Sapelo, and then I'm hoping to incorporate some barrier islands in Virginia this summer to look at that difference as well.
Very cool. Yeah, I mean, one of the things that is so cool about your research is that, I mean, you are in, let's say, 10 marshes within a week, right? Basically, 5 states within the course of, 10 days or something like that. And so, I mean, it's you get a cool perspective of how different things are. And I think that, the thing that you touched on about, basically, human population around marshes is key. And I think that, I mean, it's something that you could extend if you keep going up the coast. The raving MarshChat fans know my experience, which is, I started in marshes of the Southeast, giant untouched marshes, basically, that were, as far as you can see. And then, I moved up here, started to do research on Cape Cod and Plum Island and Nantucket. And it's essentially, kind of what you were saying about like North Carolina and Virginia, where it's like there's houses around the marsh, and marshes are a lot smaller. So many of the big cities along the eastern seaboard have been built on top of a marsh, right? Like literally the building that I'm sitting in right now was built on an old marsh, as of most of Boston. And so I think that's that's a gradient that goes all up and down the Atlantic shoreline. And I think it'll be really cool, as you continue to work on all these different sites, and as you continue to process this data, to kind of see the difference as you get, because I mean, one of the things, that kind of varies with latitude is, right, the amount of people around each marsh, right?
Yeah, so how do you disentangle that effect from just what's happening with changes in temperature or changes, commercially harvesting predatory crab species, just differences like that. Trying to wrap my mind around it all at the moment.
Yeah. Well, I mean, and you can imagine too how all that stuff is gonna change species interactions. I mean, specifically up here in New England, work from a researcher up at Plum Island. Actually, just got a job at VIMS, David Johnson, who found that blue crabs are making their way up north of Cape Cod, which they haven't either ever or in a really, really long time. And it's, a part of warming waters, that sort of thing. He has a paper about fiddler crabs doing the same thing. And, you got to imagine that as species ranges change, right, so are the important species interactions, right? As blue crabs become more or less abundant in certain areas, they're a very strong predator. So, they can have different levels of top-down control on all their different prey species, right?
Yeah, and I mean, not only is it like range shifts, but also the potential for those range shifts to also like occur alongside differences in feeding rates and body sizes and morphology, and how those to also tie into those interactions. And so you probably could expect to see a lot of variation and interactions occurring between the same species over the range.
Yeah, no, you're totally right. I mean, and I think that's one of the really cool things about your research too, is like, I mean, I think that you said this at the beginning of your talk in benthics, but basically there's no ecosystem where every single individual of the population is exactly the same size, right? And so it doesn't make a whole lot of sense to think about each of those individuals as being equal, right? And so I mean, basically, you think about this is such a cool way to think about species interactions, right? Where it's like, you have the prey species, which is a bunch of different sizes. You have the, predator species, which is a bunch of different sizes. You have the top predator species, which is a bunch of different sizes. And so, you take, your simple, snail gets eaten by a mud crab gets eaten by a blue crab, take that simple food chain and then, expand that out, based on the sizes of each one of those things. And you get potentially a completely different picture, especially when, right, like, medium-sized mud crab might eat a small snail, but he might get eaten by a medium-sized blue crab, right? Or, there's so many different chances to mix it up when it comes to size with like even the most simple sort of food webs, food chains.
Yeah, and I think that like bioenergetic demand is something that has recently, like as in the past year, been incorporated into like Lotka-Volterra, like predator-prey interactions, and it's something that people are starting to think about more as we think about a warming climate and what that means for size structure, size-specific interactions, metabolic rates, energetic demand. So yeah, essentially like what you said, one population of a predator or prey interacting with another population of predator or prey does not represent that species in a food web. Just that one particular food web at that one shot, like snapshot in time.
Yeah. So you mentioned, how this stuff might change with climate change, with a warming climate, that sort of thing. Are there like some predictions that we have right now about like how maybe, I don't know, how different like size structure might change with climate change or metabolic rate, that sort of thing? And if you're going to talk about metabolic rate and metabolic theory, explain it a little bit too for our general public audience.
Yeah, this is where I probably start to get in over my head, or I'm still working on like sussing it out myself. But I think you can go in kind of a couple different directions. If you're talking about like average body size shifts, or if you're talking about an individual versus the population. And right now I've been thinking mostly at like the level of the individual in terms of how, based on like its thermal performance curve, whether or not increasing temperature will shift it to be feeding at a higher rate or performing more optimally, or whether or not it will push it over that like optimum like thermal limit and potentially have like deleterious effects on the individual. How body sizes of an individual would respond to increasing temperature. So, and I guess an individual, over time, so not just like one individual changing its body size, but like through time. So I guess it's kind of a multi-generational process that body size would change. When you're talking about populations, I think you have to take into account like density dependence. Selection for body sizes could be driven by temperature, but it could also be driven by density of individuals within the population. And so I guess I don't know if I'm like straying away from the original question. I can't really remember exactly what that entailed.
It was basically just what are kind of the general predictions that we can make about how climate change is going to affect basically the body size dynamic when it comes to marshes or populations that have varying body sizes in general.
Yeah, and so, okay, yeah, that also depends on if you're talking about like ectotherms versus endotherms. But like, I think mostly with like these snails, for example, and with other ectotherms, the general like overarching prediction for like these marine species or aquatic species is a decline in body size, decrease in body size with temperature increases. I think there's a paper called like, naming it potentially as like the third universal response to climate change could potentially be a decrease in body size. But how that really interacts with population dynamics, I don't think has quite yet been fully explored. Yeah,
that's really cool. Well, I mean, this is all, so this is also so poignant to your research, which is like, I mean, we already kind of have a lack of understanding of what basically having body size diversity means, just like naturally, right? And so you're trying to figure that out, and then on top of it, it's like, what are the implications for as the changing climate, or as the climate continues to change, then basically from some of your research, you'll be able to make like specific predictions. Like basically, what Rebecca is going to be able to say is like, oh, when we have a population that doesn't have any big snails, this is how marsh productivity changes. This is how predation rate on that snail population changes, and then you can make potentially conclusions about how the predators will respond and that sort of thing. So I think that's one of the really cool things about your research and the population size structure stuff is that like we have this natural variation in the size of organisms right now, and it's going to be changing, right?
Yeah. Prey is going to be changing, predators are going to be changing and responding to prey at the same time. So yeah, it's a really broad question that I think probably should be tackled in pieces, but it's kind of makes your brain explode to think about it and where to start.
Yeah, well, if I know anything about your advisor, he's going to keep you broad for as long as possible.
He's doing a really good job so far.
Exactly. Oh man, that's good. Okay, Rebecca. Well, the last question of this marsh chat is, what is the craziest thing you've ever seen in a marsh?
So I was trying to think about. I was trying to go through that in my head, and I feel like I've seen a lot of. I don't know if you consider it to be crazy necessarily, but compared to what I saw as an undergrad, I've seen a lot of really exciting things in the marsh. So like I'd never seen hogs in the marsh until I went out to St. Catharines, or deer. I saw a clapper rail chick for the first time this past summer, a little black like puffball running around.
That's pretty
Adorable. Terrapins, I hadn't really seen those in the marsh before either. I think the craziest experience was actually in North Carolina when we went out, and it was there was like a lightning storm, which I wasn't expecting at the time. And it was also, okay, actually, let me take that back. Well, coupled with the lightning storm was the fact that this past summer, there were so many storms. I guess late summer, early fall. I don't know if that was like the case in the Northeast, but like South Carolina, North Carolina, Georgia were getting hammered repeatedly by these storms and crazy flood events. And so, on top of having to sample, there was a pretty consistent string of storms leading to flooding. And somebody sent me a picture of my cages that were at South Carolina, and they were actually underwater. You couldn't see them anymore. And that was like kind of the craziest thing, I think, was actually being able to visualize how high the water was getting.
Is that this picture that you shared with me?
Yeah, so those cages, that's just high tide, but they were underwater and not visible.
Oh, this is that same site?
Yeah, the same site. And so it just completely flooded. You had king tides on top of these storm events, and then fresh water just sat in the marsh for an extended period of time. And actually, when I went back to that site in late October, I noticed that the Spartina wasn't really doing as well, potentially maybe due to the flooding, and that a large number of snails had moved in. And then when I went back 3 weeks ago, it was mudflat.
Oh, wow.
Yeah, so the snails had actually, I think, coupled with that environmental stress, had grazed everything out of like that area. It was probably maybe like a 30 by 30 meter square chunk of marsh. And then the snails that were left had actually climbed into the cages that had the last bit of remaining Spartina. So densities were like 3 times as high as they should have been within those cages. And so that was shocking to me, that like over the course of 6 months, you had this environmental stress, you had these snails moving in, and then bam, like everything is gone and they've moved into the cages.
Wow, that's really cool. That's a really cool example of how quickly that die-off can happen.
Yeah, I wasn't expecting that to happen.
Yeah. So let's see, you also shared, oh wait, hang on. You also shared this picture with me.
Yeah.
What's going on here?
Pretty sure that's a clapper rail nest. We were actually walking through the marsh, and you can't see them because they're kind of nestled in between the Spartina, and they're interwoven with Spartina stalks. So we almost stepped on it, and I had to take a picture.
How big are those eggs in real life?
They're like the size of small chicken eggs, I'd say.
Yeah, I think that's about right.
They're not really tiny. They're pretty decently sized. You could probably make a good omelet with clapper rail eggs. Not that I ever advise doing that, but
of course not. But you could.
Yeah, definitely.
That's perfect. All right, Rebecca. Well, this has been a blast. Thank you so much for doing this with me.
Yeah, thank you. And I wanted to give just kind of one shout out in general to the National Estuarine Research Reserves that I've been working at. If anybody's ever like interested in like partnering with an amazing like infrastructure that's already in place and doing some really cool work, like every single near site that I've worked at so far has been absolutely incredible and super helpful. So I feel like people should take more advantage of like what's going on out there, and maybe do more partnership and more collaborative work communicating. And yeah, anyways, near sites are awesome. Had to say that. Yeah,
I mean, well, it's so great, and it works so well for your research because there's a salt marsh National Estuarine Research Reserve, all up and down the coast. So you can just pop in and measure the latitudinal variation, and that's that.
Yeah, exactly. It's just right there. It's so perfect.
That's awesome. Well, thank you so much, Rebecca, for joining us. Please tune in, I think either in next week or a week after, we will have David Johnson on MarshChat, which should be a lot of fun. So thank you, everybody, for coming and joining MarshChat. Bye-bye.
Thanks, Mark.